This is Dr. Saad in front of you, and from today onwards we will be discussing a very important topics of the neoplasia. So first of all is the hallmark of the cancers, means that what changes in the cell may occur that it may become a cancer cell, or what are the properties of the cancer cells. These are the hallmarks. So there are 8 hallmarks and we will be studying these hallmarks in detail one by one, but here I am giving a brief introduction about these all 8 hallmarks. Clear? So starting from the first hallmark, that is the self-sufficiency in the growth signals. It means that the tumor cells, they are not dependent upon the external stimuli, means they have self-sufficiency in growth, means they do not need any stimuli, any external stimuli or any internal stimuli to carry out the growth. Without the stimuli they can carry out their growth. This is self-sufficiency in the growth signal. Then we have the insensitivity to the growth inhibitory signals, means that there are any stimuli that causes or any substance that inhibits the proliferation, inhibits the growth, that substance cannot inhibit the growth in the cancer cells. Clear? That is not effective. Insensitivity means it is not responsive to the growth inhibitory signals. Simple as that. Then we have the altered cellular metabolism. It means that it carry out the aerobic glycolysis, that is called as the Warburg effect. We will be discussing each and every hallmark in detail, but I am telling you just a brief introduction so that you may be aware about their names. Clear? Then we have the evasion of the apoptosis, means that the cancer cells also must evade, means resist the apoptosis. Then we have the limitless replicative potential, means that normal cells, our normal cells, they have a limited capacity to divide, means every cell, for example, for suppose a cell have a capacity to divide about 60 times. After dividing 60 times, that cell will become aged and it cannot replicate. But in the cancer cells, what happens that they have limitless, means unlimited replicative potential, means they can replicate unlimited times. That is called as the immortality, means never dying. Clear? Then we have the angiogenesis, means obviously the cancer cells, they have to grow, and in order to grow they have to means supply the nutrients, and they also need oxygen. So for this purpose they also carry out the growth of the vessels, that is called as the angiogenesis. Then invasion and metastasis we have already discussed in detail. Then we have the evasion of the host immune response. Evasion means resistance, means obviously if the cancer cells they start to develop, there will be changes in our body and the body will respond, means our host immune response will be activated. So the cancer cells must resist to that host immune responses. These are the 8 hallmarks. We will be discussing each and every hallmark in detail, and starting with the first hallmark, that is the self-sufficiency in the growth signal. So I am erasing these 8 hallmarks. This was just a brief introduction about these hallmarks. Now we will be studying the first hallmark, that is self-sufficiency in the growth. I have already told you that we have the certain genes that promotes the growth, and that are called as the proto-oncogenes. Clear? We have studied the proto-oncogenes. These are the normal genes that carry out the growth, but the mutation in the proto-oncogenes, now these becomes the oncogenes. Whenever the mutation occur in the proto-oncogenes, they become the oncogenes, and when these oncogenes they produce the proteins, they form the proteins, that are called as the oncoproteins. Clear? Now those oncogenes, those oncoproteins, before studying these, we have to study the normal pathway of the growth, normal genes that are involved in the growth. So first of all, moving on towards the normal pathway of the growth. What is the normal pathway? Simple is that, for example, you have a stimuli. This is the stimuli, you can say this is a growth factor. Clear? This is a protein. Clear? That protein, it is having a receptor, means it is attached to a receptor, and that receptor is the having a cytoplasmic domain. Clear? This is your, for example, stimuli, this is your receptor, and this is your cytoplasmic domain. Now a growth factor comes, attaches to the receptor and activates the internal protein. These are, you can say, this cytoplasmic domain, it is associated with a protein here. This protein becomes activated, and now this protein, it activates the signal pathway, signal transduction pathway, or you can say second messengers. For suppose this is your nucleus here. Here you have to move those proteins. Now these proteins activate the signal pathway or second messenger that move towards the nucleus, and in the nucleus obviously what will be carrying out, they will be activating the genes. The genetic material is present here, so genes will be activated and transcription will be activated, because we have to form the proteins. In order to carry out the growth, we have to form the proteins. So these genes are activated, transcription occur, then the translation occur, and finally after here there will be, you can say here, transcriptional factors, they are also present. And now finally after transcription and translation we have the proteins, and now these proteins will be helping in the cell cycle progression. Clear? Now the cell cycle will progress. So this is a normal simple pathway that you all, I think so, you all know about this pathway. Now there are different signaling pathways that we will be studying. For the most important pathway is the tyrosine kinase pathway. I think so you have already listened about this pathway. There is tyrosine kinase pathway, G coupled protein pathway, we have the WNT signaling pathway, we have hedgehog pathway. Clear? So we have different pathways, different signaling pathway, but the most important pathway is the tyrosine kinase pathway. And why this is most important? Because this is the most common pathway that is mutated, and that is the most common pathway which is the most common cause of the human neoplasm. Clear? Most common cause of the human neoplasm. That's why we will be studying this tyrosine kinase pathway in detail. Clear? So first of all, here I will be studying the tyrosine kinase pathway. For suppose now I am explaining you the normal tyrosine kinase pathway. If we will be studying the normal pathway, then we will be moving on towards the mutation in those genes that are involved in this pathway. So first of all, this is your growth factor. Clear? It comes and attaches to the receptor that is called as the growth factor receptor. This is your growth factor receptor. Now this growth factor receptor, it is associated with a protein here, and now this protein is basically called as the RAS protein. Clear? RAS protein. This RAS protein is basically in the inactive form. This is now inactive. For suppose this is a inactive, and inactive form, it is associated with the GDP. Clear? It is associated with the GDP. Now then what happens, that the signal come, growth factor come, and acts on the receptor. This receptor activates this protein. Now the RAS protein, after activation, this is coupled with GDP. This is coupled with GTP. Clear? Now it becomes activated. For suppose now I am writing here, this is activated and this is inactive RAS protein. Clear? Now this is activated. This we are studying the tyrosine kinase pathway. Clear? Now after activated means it becomes coupled with the GTP. Now this activated RAS protein, it carrying out the, means it produce two downstream pathways. So what are two downstream pathways? Number first is the, here is RAF, then we have the phosphoinositide 3 kinase. You don't need to remember the full form of these genes or the proteins. No need to remember them. Just it is fine to remember, just to recognize them. Clear? Then this after RAF, it activates the MAPK, mitogen activated protein kinase. Clear? These are the enzymes. Clear? Now this I already told you that there are certain cytoplasmic proteins that move towards the nucleus. Now from here MAPK, it will be moving towards the nucleus in order to carrying out the activation of those genes, the genetic material. Now MAPK, it will be carrying out the, or you can say activate the transcription. Clear? And in the transcription obviously we have certain transcriptional factors. So what are those? Those are the D-cyclins and the MYC. You can say these are the transcriptional factors. Clear? This is the one downstream pathway of the RAS. The second one is the PI3K, phosphoinositide 3 kinase, and this activate AKT. The AKT has means act on different, you can say, substrates. One of those substrate is the mTOR. mTOR. Clear? Then okay, after this transcription, what happens that they obviously will be carrying out the protein synthesis, or you can say then what happens, then cell cycle progression occurs. Clear? These D cyclins MYC and mTOR, they all carry on the protein synthesis. In this way the cell cycle progresses, and in this way the growth occurs. Clear? So in the further lectures we will be studying about the mutation in these genes, means mutation in the RAS, mutation in the MAPK, mutation in AKT, so mutation in mTOR. These all the mutations we will be studying. And remember one thing, that these all are what? These all are the growth promoter genes. And I have already told you in the previous lecture that the growth promoter genes, which type of mutation must occur in order to produce cancer. There is a gain of function mutation, very important. This is very important to remember, that all of them there will be the gain of function mutation, because they are all what? They are all growth promoter genes. Clear? This concept is already cleared in your in previous lectures. Clear? So this is your whole pathway, normal pathway of the tyrosine kinase pathway, and in the further lectures we will be studying about the mutation in these pathway. Clear? So if you have any confusion, any problem, you may ask me in the comment section. And thank you so much. Allah Hafiz.